Welcome to the knowledge domain, the concept of life sciences and biotechnology. In today's lecture, we are going to learn a brief idea about the taxonomy and we will discuss in detail the various criteria of naming an organism. So the taxonomy originated from the Greek word. The first part is the taxis, which means arrangement, and the nomia means method. So it is a arrangement method. Next is the nomenclature. It is from the Latin. Nomen means name and calere means to call or to call someone with a name. That is the nomenclature. So what are the criteria of binomial nomenclature? It has to be unique. There should be uniqueness. That is published in the Systema naturae in the 1758. Proposed by the Carolus Linnaeus in the 1030s. There should be the universality, means everybody in the world should accept it, should use it, and it should be stable, means it should not change time to time. So these are the three important criteria to give a binomial name to someone. Okay. So the first classification of the entire living organisms were done by the Carolus Linnaeus in 1735. This is called the two kingdom classification. The one is the vegetabilia and the second one is the animalia. Then it is slightly improved by the Haeckel in 1866, who improved it by another kingdom and called it the three kingdom classification by including Protista, which is a means unicellular organism. Then Plantae Animalia, as discussed by the Carolus Linnaeus. Followed by the Haeckel, it is the Whitaker, which was the most accepted one till the recent time. That is proposed in 1969 and is five kingdom classification, where the Monera, the prokaryotes are separated from the rest of the eukaryotes. Then Protista are the unicellular eukaryotes, then Plantae, fungi, and the Animalia. Then around 1990, Woos, along with some other workers, they proposed a three-domain classification, and each domain is further divided into the kingdom and the phylums that we will discuss. So the three domains are the bacteria, archaea, and the eukarya. This is also called the eukaryotes. This is based on the ribosomal RNA characters of the various organisms. And in case of the three-domain classification, they also include the cell membrane lipid structure and the sensitivity to the antibiotics. So, entire living world is originated from a common ancestor that is called the last universal common ancestor LOCA that gives rise to the three types of the cells. And based on that three types of the cells, we have the bacteria, archaea, and the eukarya. And these are the characters of the archaea, that the membrane hydrocarbons are branched and it is attached to the glycerol by the ether linkage, whereas in case of the bacteria and the eukarya, it is the ester linkage. In archaea, there was no peptidoglycan. In bacteria, there was peptidoglycan. And archaea is sensitive to some of the antibiotics. Those are also anti-eukaryote, means the eukaryotic cells are sensitive, but it is completely insensitive to the antibacterial antibiotics. And the examples are the methanogen, extremalophiles, and the hyperthermophiles. So what is the characters of the bacteria and the eukarya? One of them, bacteria has a cell wall. In case of the eukaryote, the plant cells have cell walls. And bacteria, there the peptidoglycan, as we have discussed. And the membrane lipids in the bacteria and the eukarya, both are the unbranched membrane lipids. Bacteria are sensitive to antibacterial antibiotics and insensitive to the anti-eukaryotic antibiotic, where the eukaryotes are sensitive to anti-eukaryotic antibiotic, whereas less sensitive or no sensitivity to another antibacterial antibiotic. Okay. The example of the bacteria, the mycoplasma and the cyanobacteria, as well as we know there are gram-positive and gram-negative bacteria. And this eukaryote or the eukarya is further subdivided into the four kingdoms. Those are the protista, which are the unicellular eukaryotic organism, and the fungi, who cannot synthesize the carbohydrates, so they are the heterotrophic multicellular eukaryotes. Then we have the plantae and the animalia. So this is based on the ribosomal RNA sequence, and this the phylogenetic tree that is prepared from the ribosomal RNA. We have the three domain classification. So what is a phylogenetic tree? Phylogenetic is based on the phylogeny, which gives us evolutionary relationship between the organisms. Okay. So in the taxonomy, there are two other terminology. One is the alpha taxonomy, that is the discipline which deals with finding, describing, and the naming of taxa, particularly the species. It was proposed by the William Bertram Turrell. Then Ernest Mayr proposed the beta taxonomy in 1968 that deals with the classification of the ranks higher than the species. So when it is related to the species, naming species, finding and describing them, it is the alpha taxonomy, and when it is higher up in the order, it is the beta taxonomy. So there are the salient features of the code based on the book published by the Linnaeus, Systema Naturae. The 10th edition, it is properly described. So the course, this book has a preamble. It has 86 articles, 5 appendices, and a glossary and a detail index. Okay. So the year it was published at 1750. So what are those codes? The name in the binomial nomenclature must be Latin, or if it is not Latin, it has to be Latinized. Names of the taxa higher than the species should be uninominal, and the name of the species is binomial. So when it is a name of a species, it is binomen. And there are two names, and all the names higher up in the order are the uninominal. And if there is a subspecies, it is a trinomen. So there are three names of a species. So names of subgenus is placed in parentheses between the genus and the species, like your Saurides, Gonophonus niger. So Gonophobus is the subgenus that is in the parentheses between the genus and the specific name. And the family name should end in DA, like the Tupinidae. So all the family ends with the DA. Genus name should be a noun in nominative singular or related, treated as such, like the Epicena. So genus is a noun in nominative singular. While the species name can be an adjective, it can be a nominative singular like the genus, but it agree in gender with the generic name. For example, the Drosophila obscura or the Felis tigris. Or it can be a noun standing in opposition to the generic name like the Felis leo. So genus is always a noun nominative singular, whereas the species name can be an adjective. It can be like the genus nominative in singular, or it can be noun, the species. Species can be the three forms of the figure of speech. So the name should be independent of the other system, which should not depend on the other in naming a species. All names given to the species from time to time should be mentioned in the synonymy. So like one species name is continually in the optimization, so all the names should be there in the synonymy. Author name should not be part of the name, but it can be used as an optional and is suffix, like the cancers, Pegaros, Linnaeus. So it is suffix and it is optional. It is not mandatory. Author name. Then there should be the law of the priority, means we should prioritize it. The valid name is the oldest name published and available. So if there is a name, so the valid name should be chosen, the oldest name available, or it is published, depending on which one is earlier. Then there is the synonymy, that one we have came across here. Different names assigned to the same taxon, they should be mentioned along with the valid taxon, like the areas Vitella or the areas Fabia. So they are the two names, Vitella and the Fabia. So it should be mentioned along with the valid taxon. Synonyms are the different names to the same taxon. Then there is the homonym, identical names in spelling for different species of the same genus and for different genera of a family, and it is always rejected, because if there is a homonymy, then it cannot be, we cannot identify or characterize any species, so it is rejected. But in case of the synonymy, they are placed mentioned along with the valid taxon. Next, the holotype. It is the single specimen. So when we name someone, it has to restore the representative specimen. So holotype is the having the most number of feature of a species, that is considered as holotype. So it is a single specimen on which description of the species is based, and it is red color level is fixed on the specimen. But the allotype is the opposite sex to the holotype, also carry the red level. So holotype is the representative specimen, allotype is the opposite sides of the representative specimen. Then there is a paratype. So if a investigator or the researcher, they found lot of species and most representative one is kept as holotype, and from the opposite sex one allotype is kept, the remaining all specimens are the paratypes. So after designating one specimen as the holotype, another as the allotype, then the remaining all assigned specimens are the paratypes, and they are the yellow level. So holotype, allotypes are the red level, and the paratypes are the yellow level. Next is the syn type. If no holotype is designated, suppose the researcher did not find any specimen that properly characterized, means one specimen properly characterized the entire organism. So in that scenario, specimen that author studied for the description of the specimen is the syn type. So the specimen that is studied means for the description of the species is the syn type. So lecture type, in the absence of the holotype, any of the syn type can be designated as a lecture type. So it is a type of syn type, holotype, when there is no proper representative. So the one specimen from the syn type, we properly represent the organism will be tagged as lectotypes. So this is only given when there is no holotype specimen. So like paratype here, this is paralectotype. So the remaining all specimens are the paralectotype. And this is the neotype. If all type specimens are destroyed, a new type that fits the description very well, that will be tagged as the neotype. And this is in the exceptional circumstances. Thank you.